Hello everyone, welcome to this first lecture on basic concepts of genetics. In today's lecture we will see some basic concepts regarding DNA and RNA. Now what is genetics? Genetics, it is a study of inheritance and variability of traits. Traits, that means characters. Now there is difference between these two terminologies. Inheritance, which means transmission of genetic information from parents to offsprings. It is basically a vertical gene transfer. On the contrary, variability means changes due to some genetic alterations or some environmental factors. So this variability can result from either recombinations or horizontal gene transfer, or it can also occur due to mutation, which is sudden change in gene. So what is genetics? It is a study of this inheritance, which is transfer of genetic information from parents to offsprings, and variability. Variability means changes in genetic alterations. Now when we talk about microbial genetics, you will come across a scientist, Gregor Mendel. He is referred as father of genetics. What did he do? He performed experiments on pea plant. He studied various traits of that plant, color, size, shape, and he come across law of heredity. So these laws of heredity, these are discovered by Gregor Mendel, and those are universal to all life form. It means to plants, animals, bacteria, and whatever study he performed or his results, he published those in experiments with plant hybrid 1865. What is the importance of microbial genetics? Why studying microbial genetics is important? Because genetic principles, these are universal, and when we say universal, our microorganisms or microbes, they also follow these universal principles. Actually, in the beginning, scientists were reluctant to use microorganisms for studies of genetics because they used to think that these microorganisms are too small and they might not contain systems which are present in eukaryotes. But studying microorganisms has its own benefits, and as a result, after several experiments, now scientists uses these bacteria and viruses as major tools for genetic research at molecular level. What are different benefits of using these microorganisms for study of genetics? These microorganisms, this bacteria, it contains millions of cells, and because of this, it allows the discovery of rare genetic events through selective techniques. Another advantage of using bacteria or microorganisms for genetics study is that they are having a single chromosome. Because of this, mutation is immediately visible. That is, no masking occurs when these microorganisms are used. If they contained multiple chromosomes, then there were high chances of masking effect, and some mutations might have gone unnoticed. Another advantage is microorganisms are rapidly growing. They have a rapid growth rate, and therefore quick experimentation and observation is possible in case of microorganisms. Next advantage is they can be easily cultivated. In microbiology laboratory, cultivation is too common. What we do, we only provide certain optimized condition. Certain controlled condition is provided, environment is created in such a way that that particular microorganisms will be quickly grown. That is the only thing they will need. They will need nutrients and they will need favorable conditions. So this easy cultivation is another advantage of using microorganisms in genetics. And last one is metabolic diversity. When we study types of microorganisms, some are mesophilic, some will require lower temperature, some will require higher temperature. That is, there is a huge diversity among these microorganisms. And because of this, wide range of study opportunities and research is possible when these microorganisms are used. So let us now see DNA. DNA, as you know, it stands for deoxyribonucleic acid. It is a polymer of deoxyribonucleotides. It is usually double-stranded. It has a double helix structure. It is found in chromosomes, mitochondria and chloroplast in case of eukaryotes. And it is found in nucleoid in case of prokaryotes, as it does not have a membrane bound nucleus. It acts as a genetic material in most organisms. Now here most organism is said because in case of viruses, RNA is present as a genetic material and DNA, it carries genetic information. Now key events in the discovery of DNA structure. Friedrich Mischer in 1868 found an acidic substance in nucleus to which he called as a nuclein. He did not term DNA to that substance. On the contrary, he called it as nuclein. So you might come across a question on who discovered an acidic substance called nuclein. So answer is Friedrich Mischer. As we all know, in 1953, a pair of scientists named James Watson and Francis Crick, they proposed the famous double helix model of DNA. Now let us see some of the historical experiments. Those proved that DNA is a genetic material. First comes Griffith's experiment. In 1928, Griffith found a transforming principle to which he called as transforming substance. What did he do? He studied Streptococcus pneumoniae, which is a causative agent of pneumonia that causes pneumonia disease. So there are two strains of this Streptococcus pneumoniae. One is S strain, another one is R strain. Yes, as the name suggests, it is a smooth strain. R strain, as that R indicates, it is a rough strain. Now why it is called as smooth and another one is called as R strain? Because in case of this Streptococcus, there was a capsule present around one strain and that gave it a smooth appearance, and because of this capsule it was virulent. It was able to cause disease. Another one, as the name R strain, rough strain, it was not having this capsule and thus it was non-virulent. So what did this Griffith do? He injected this smooth live strain of Streptococcus pneumoniae in mice and he found that mouse died. Then he injected mice with live R strain. As it was rough strain, it was non-virulent, mice survived. In third case, he injected this heat-killed S strain. He took live strain, he heated that strain, and these heat-killed strains were now injected into a mice, and to his surprise, mice survived. And finally, he injected mice with heat-killed S strain and live R strain. Now, when the heat-killed S strain was injected into a mice, that mice did not die. So, when that heat-killed S strain along with R strain itself was non-virulent, this heat-killed S strain was again non-virulent. So, when he combined these two strains, injected it into a mice, he found that mice died. Mice could not survive. So, he came to a conclusion that there was something which transformed this R strain, live R strain, into a virulent strain, because R strain itself was non-virulent. S strain was virulent, but as it was heat killed, he thought that there was something present in this S strain which transformed this R strain into a virulent strain, and he called it as a transforming principle or transforming substance. He could not name it as DNA. He did not know that it was a DNA. Now comes Avery, MacLeod and McCarthy. They found that Griffith's transforming principle was nothing but a DNA. In 1944, they performed an experiment. Now how did they perform this experiment? It will be another lecture, but due to time constraint, I will cover it in short, and I recommend you to go and study this experiment because it is very interesting experiment to study. They extracted molecules from that heat-killed S strain. They extracted proteins, DNA and RNA. How did they extract it? They extracted it with enzymes, treating it with enzymes. They mixed treated extracts with live R strain, and they found that transformation occurred when these protein and RNA were destroyed. But when they destroyed DNA, no transformation occurred. And thus they came to a conclusion that DNA is a substance that carries genetic information. And thus they said that it was DNA which Griffith found as a transforming principle. Now remember that again the Avery, MacLeod and McCarthy could not came to discovery of DNA as a genetic material. It was this pair of scientists, Hershey and Chase, who found that DNA is a genetic material. They performed an experiment with bacteriophage T2 Now, as you know, bacteriophage is a virus which infects bacteria. So this T2 infects E. coli. In 1952, you have to remember scientist's name, what experiment they do, and what was the year in which that experiment was conducted. All these are important for your examination. So in 1952, Hershey and Chase performed an experiment with bacteriophage T2. There they used radiolabeled elements. They used 32P and sulfur with 35. Now, as you know, DNA, it contains phosphorus, whereas sulfur is present in protein coat. So they performed this experiment again. I recommend you to go and study this experiment. So they found that when this radio-labeled DNA with radioactive phosphorus and labeled protein coat with radioactive sulfur, when that was treated on bacteriophage, and that bacteriophage is now allowed to infect bacteria. Then they separated phage coats from bacterial cells. They lysed that cell, studied its protein coat and its genetic material, and they found that the sulfur, it stayed on outside protein coat as it was on protein coat, whereas this radiolabeled phosphorus, it entered bacteria. As a result, they came to a conclusion that it was not a protein but it was DNA, which is the genetic material that directs virus replication inside a bacteria. When we talk about DNA structure, we only talk about Watson and Crick, but there was this scientist who was responsible for discovery of this DNA structure as much as Watson and Crick were. Now let us see who this scientist was. Her name was Rosalind Franklin. She worked in the same laboratory as Maurice Wilkins, and she used X-ray diffraction study to see what this DNA structure, what these DNA fibers are like. So she used X-ray diffraction study, and some major advances in X-ray diffractions were made, and by her study she came to certain conclusions. She found that DNA is not a single stranded. Until that, scientists were only thinking from one perspective, that DNA is a single stranded, but her X-ray diffraction study showed that DNA contains more than one strand. She also found that DNA is a helical structure, and she also found one turn of DNA contains 10 base pairs. So these were major findings. Those were later used by Watson and Crick to come with that DNA double helix structure. So what is this DNA structure looks like? It contains phosphorus, it contains sugar, which is deoxyribose in DNA, and it is ribose in RNA. This is 5 carbon sugar, deoxyribose sugar, phosphate, and nitrogen base. So DNA structure, it is described at 4 levels. It contains deoxyribose sugar, pentose sugar, nitrogen bases, which are 4 types, ATGC in case of DNA and AUGC in case of RNA, and a phosphate group. So this whole structure is a nucleotide. Nucleotides, these are the building blocks of DNA. So what does the nucleotides look like? It is a nitrogen base plus sugar plus phosphate group. When it lacks phosphate group, we call it as a nucleoside. So nucleoside is a nucleotide without phosphate group. What are nucleotides? They are formed by condensation of sugar, phosphate, and one nitrogen base. Now that nitrogen base can be adenine, that nitrogen base can be guanine, that nitrogen base can be cytosine, and that can be a thymine in case of DNA. When that adenine is paired with ribose sugar by glycosidic bond, we call it as adenosine. So we call it as deoxyadenosine in case of DNA, adenine plus deoxyribose. When it pairs with phosphate, we call it as a deoxyadenosine monophosphate. If there are 2 phosphates, we call it as diphosphate. If there are 3 phosphates, we call it as ATP, deoxyadenosine triphosphate. So here are DNA nucleotides and RNA nucleotide structure. Let us see what are the difference between these two structures. On this second carbon, in case of deoxyribose, there is instead of hydroxyl group, only hydrogen moiety is present. Whereas in case of ribose, on this second carbon of ribose sugar, OH group is present, hydroxyl group is present. Now remember that this second hydroxyl group makes this RNA unstable structure. How? Because this hydroxyl group is acting as a nucleophile. That means it will act as an electron donor. It will more like acting as a knife which is ready to attack on a molecule. So it attacks on this phosphate group which will be pairing with another nucleotide. So this phosphate group is attacked by this second hydroxyl group which is present on that ribose sugar. As a result, RNA is not stable as compared to the DNA. It makes it reactive. This OH groups makes it reactive. Another difference is thymine is present in DNA whereas uracil is present in RNA. So this is nucleotide in DNA and this one is nucleotide in RNA. This phosphate group is linked to ribose sugar in both the cases on this fifth carbon of ribose sugar by phosphoester bond. And nitrogen base, whatever that nitrogen base, it can be AGC or AUGC. It will be attached on the first carbon of ribose sugar by glycosidic bond. These nitrogenous bases, they are divided into two groups, purines and pyrimidines. What are pyrimidines? They are made up of 6-member ring. In case of purines, it is again 6-member ring, but it is fused to form a 5-member ring. So you might have a question on which of the following contains 2 rings. So these 2 rings structure is present only in purines, A and G. What are purines? Adenine and guanine, they are purines. Whereas thymine, cytosine and in case of RNA, uracil, these are pyrimides. So pyrimides are single ring structure whereas purines are double ring structures. These rings are not only made up of carbons, they also contain nitrogen. The nitrogen bases, these are double ring structure in case of purines and single ring structure in case of pyrimides. Here you can see a uracil which is present in RNA and here is a thymine which is found in DNA. What is the difference between these two structures? On 5th carbon, methyl group is present in case of thymine whereas it is absent in uracil. So you have to remember that uracil is a nitrogen base which is found in RNA whereas thymine is a nitrogen base which is found in DNA. So these nucleotides which are building blocks of DNA, they are linked to each other by phosphodiester bond. Now why it is called as phosphodiester bond? Now let us see this structure. Here it is binding with this sugar with phosphoester bond and here it is binding with third carbon of another deoxyribose sugar again with phosphoester bond. So this phosphate is diesterified and that's why this complete bond here, this bond will be called as phosphodiester bond. Here it is bound with phosphoester bond. Here again it is bound with phosphoester bond which makes 2 phosphoester bond and thus it is called as phosphodiester bond. So DNA, it is a double helix structure. That means there will be 2 strands which will be paired with one another with nitrogen bases. Now how these nitrogen bases are paired? They are paired with hydrogen bonds. Purine pairs with pyrimidine, purines, adenine and guanine, they will be pairing with respectively thymine and cytosine. So AG, they are purines, CT, they are pyrimides. So purine A pairs with pyrimidine T with 2 hydrogen bonds, whereas guanine will be paired with cytosine with 3 hydrogen bonds. Now as RNA does not contain thymine, there will be presence of uracil. This uracil will pair with adenine with 2 hydrogen bonds. Now there is one rule in case of double helix structure and that rule is called as Chargaff's rule. What is that Chargaff's rule suggest? It suggests that percentage of purines will always be equal to percentage of pyrimidines in case of double helix structure. That means as much as there are A, there will be that much T's in case of one structure, because in one strand A pairs with T of other strand. So जितने एक स्टैंड में एडेनाइन होंगे उतने ही उस डीएनए में डबल हेलिक्स स्ट्रक्चर में थायमिन होंगे, जितने उसमें ग्वानाइन होंगे उतने ही उस डबल हेलिक्स स्ट्रक्चर में साइटोसिन मिलेंगे. बट रिमेंबर दैट दिस A plus T and G plus C ratio that varies between species. Let us see helical parameters of DNA. Here we are talking only about B form of DNA, which is the most common form of DNA. It is a right-handed helix. It twists clockwise And if we see this one turn of DNA, let us see this structure. Okay, let us see this structure. Here A and another A is present on this strand, so distance between these two nitrogen base is 0.34 nanometer. If we convert it into angstrom, it will be 3.4 angstrom. This complete turn of DNA will be having 10 nitrogen bases, so it will be around 34 angstrom unit distance. If we talk about this helical structure, it contains some grooves. Now there are two types of grooves are present, major groove and minor groove. Major groove is a wide and deep, whereas minor groove is a narrow and shallow. Why these grooves are important? These grooves are important because here protein will be acting on that DNA strand and it will recognize these grooves for binding of DNA. These are the different forms of DNA, A, B and Z. B form of DNA is most common. It is right-handed. A form is also right-handed. Z form is left-handed. See diameter. Diameter is around 20 angstrom, 2 nanometer in B form of DNA, whereas this A is wider, diameter is larger than B form. Diameter in case of Z is smaller than B form. Per turn, 10 bases are present in B form, 11 bases in case of A form and 12 in case of Z form. So these are differences. You can pause this slide and study these differences among these forms of DNA. But remember, most common form of DNA is B form. Now let us talk about DNA supercoiling. Why this supercoiling is important? Because DNA contains around 1.6 million base pairs. If that DNA is straight, it is not possible to fit it into a bacterial cell which is barely a 1 to 2 micrometer in length. So to fit itself into a bacterial cell which is such a small structure, this DNA supercoiling is important. So what this DNA supercoiling is, it is over and underwinding of DNA strands. It is important for DNA packaging since DNA is much longer than cell, prokaryotic cell. यूकैरियोट्स के अगर हम बात करें तो वहाँ हिस्टोन प्रोटीन्स होते हैं जिसके अराउंड डीएनए वाइंड होता है और वो फिट होता है. In case of bacteria or prokaryotes, histone proteins are absent. Okay, so topia isomerases are enzymes. It contains enzymes such as DNA gyrases. It is a class of enzymes which contains enzymes like DNA gyrases in bacteria which are responsible for supercoiling of DNA. As a result, the supercoiling reduces space and it allows compact packaging of DNA inside a bacterial cell. Now let us talk about RNA. So history of RNA in brief. We have talked about Frederick Mischer. These RNAs as a genetic material were discovered by certain scientists, of which first is Ivanovsky. Dmitry Ivanovsky, who studied tobacco plant in 1892. He found that a sap from infected tobacco plant remained infectious even the bacteria were removed from that sap. He could not find what that sap contained. Okay, so in 1898 Bejernik studied that sap and he named it as a contagium vivum fluidum. There is certain element, certain fluid, certain substance in that sap is responsible for infection of healthy plant. It was the first discovery of virus. So virus was recognized firstly by Bejernik, but he could not crystallize it. It was Stanley who crystallized that tobacco mosaic virus and showed that virus contains proteins and RNA. Then Harris and Chase, they are credited for their study on bacteriophage MS2. Here they found that RNA alone is a genetic material. They studied that in tobacco mosaic virus, RNA was a carrier of genetic information. It was a genetic material in TMV. Now in 1961 genetic code was deciphered. Nirenberg and Mathai, they for the first time found a codon first codon was decoded, and they confirmed that RNA codons specify amino acids. Codon, there are 3 nucleotides present on mRNA which codes for amino acids. Then in 1970, an important experiment was carried out by Girer and Sham. They purified tobacco mosaic virus RNA that alone infects tobacco plants, and they came to a conclusion that RNA is sufficient enough genetic material, and it is only genetic material, and this proton coat did not have any role in case of infection. Then came scientists Pear, Temin and Baltimore. Around 1970, they discovered that reverse transcriptase enzyme is an enzyme which produces DNA from RNA. Until then, DNA to RNA central dogma was only known, but they found this enzyme which forms DNA from RNA. And they also found that RNA serves as a genetic material in retroviruses. So if we talk about this structure of RNA, it is ribonucleic acid. It is a polymer of nucleotide similar to DNA. It is found in all living forms and some viruses. Now what is structure of this RNA? Basically they are required for protein synthesis, regulation, and in case of certain viruses, they act as a genetic material. So usually RNA are single stranded, and DNA, they are usually double stranded. So being single stranded, that also makes them unstable, but they can form secondary structures like loops and hairpins. As the tRNA, it is having loops and hairpins. Structure of RNA, like DNA, it contains a sugar, ribose sugar, 5 carbon sugar, nitrogenous bases A, G, C, U, adenine, guanine, they are purines, and uracil and cytosine, they are pyrimidines in RNA, and it contains phosphate group. Types of RNA: mRNA, rRNA, tRNA, they are most common. M stands for messenger RNA, T transfer RNA, and R ribosomal RNA. What are their roles? mRNA, they acts as a template for protein synthesis. tRNA, they brings amino acid to ribose during translation, and rRNA, they have their role in protein synthesis. This mRNA carries genetic information from DNA to ribosome. tRNA, it is clover-shaped molecule. Hairpin model is also famous in case of this tRNA. rRNA, it is structural and catalytic component of ribosome. sRNA, it is a small regulatory RNA which is having a short non-codon RNA, and it regulates gene expression. Apart from these common RNAs, there are certain viral RNAs which can be single-stranded, positive sense or negative sense, or double-stranded RNA, and those acts as genetic material in RNA viruses. For example, in TMV, tobacco mosaic virus, it contains RNA as genetic material, whereas adenovirus, it contains DNA as genetic material. So in case of viruses, they will contain either DNA or RNA as their genetic material. But if we talk about prokaryotes, other prokaryotes like bacteria, bacteria will always have DNA as their genetic material. Now let us come to see certain terminologies which will be asked in your examination, and those terminologies are codon, anticodon, and codogen. So what is this codon? It is a sequence of 3 nucleotides. What are nucleotides? We just saw in this lecture. So these 3 nucleotides on mRNA are termed as codon, and their role is in the coding of certain amino acid. So let us see, this is mRNA. Here it will have its 5' end, here it will have its 3' end. So let us see, there are 3 nucleotides, A, U, G. These A, U, G, these are nucleotides on mRNA. So they will code for methionine. What is methionine? It is an amino acid. And these A, U, G, they are termed as start codon. Each codon is triplet based. It will always contain 3 nucleotides, and there are 64 possible codons. It includes start codon, and there are certain codons such as UAA, UAG. These 3 codons are called as stop codon. Why? Because when these codons are present on mRNA, there are no amino acids which are coded by these codons. As a result, they are called as stop codon. So you will be asked in examination for start codon and stop codon. So what is the role of this codon? It provides genetic code for protein synthesis. Where is it present? It is present on mRNA. So remember these points. Now let us see codogen. Codogen, it refers to the gene or DNA sequence. Whatever gene sequence is present on DNA, it is called as codogen, and it encodes a specific polypeptide. In other words, to simplify this terminology, it is a segment of DNA whose nucleotide sequence determines the codons on mRNA, which then specifies the sequence of amino acid in protein. Example, gene coding for insulin is codogene. Function is blueprint for RNA. So codogen, it will be terminology related to DNA. Codon is related to mRNA. And anticodon. Anticodon is a sequence of 3 nucleotides. Again, it is a triplet nucleotide, but on tRNA. Codon was on mRNA. Anticodon will be on tRNA. And it is complementary to a codon on mRNA. So remember that example. AUG, which codes for methionine, it will be having a complementary pair on tRNA. So on tRNA, it will be UAC. So this is mRNA codon AUG. It will be having its complementary pairing of UAC on tRNA. What is the function of this anticodon? It ensures that correct amino acid is added to the growing polypeptide during translation. It is found on the tRNA molecule. What is the role of this tRNA molecule? tRNA, it is transfer RNA. It transfers specific amino acids. So this was all about basic concepts of DNA and RNA. Hope you have understood this lecture. In case of any queries, you can ask that in comment section. Thank you.